Thank you. Yes, thank you, and also thanks for the invitation. Yeah, it is this, I guess you can call it ancient DNA, but I think the other joke was this is also quite an old project, which is actually absolutely true, because we actually started this, I think the initial sequencing runs were back in 2020. Of course, in hindsight, had we had the review, we probably would have finished a lot sooner, but at least we did finish. Actually, we just finished a few last month. So we're very busy with the data analysis. But what I'm going to be talking about today is the fact that we've just finished sequencing 500 human genomes, split evenly between Alzheimer's disease patients and cognitively healthy centenarians. And as I mentioned, we're busy with the data analysis, but I'll give you a couple of snapshots of some of the things that we've been doing on structure variations, how they can change methylation patterns, and then echoing also some of the discussions from the panel discussion session. Also ancestry, right? Because it's absolutely true that we're completely bad when it comes to looking at genetics outside of European individuals, especially when it comes to genetic risk factors. So I'll talk about at least a little bit of an effort into trying to at least contextualize some of this finding in diverse populations. So to start, just want to give you a bit of a background. So our group is interested in looking at genetic risk factors in dementia. And that's because we know that with aging, some people will unfortunately go on to develop neurodegenerative diseases, including Alzheimer's, Parkinson's, and several others. And although the environment, there we go. Although the environment does lead to, can influence our risk for these neurodegenerative processes, genetics is a major risk factor. For example, in Alzheimer's disease, which leads to the most common form of dementia, we know that between 60 to 80% of the risk can be genetic. Now, Jonas had a nice introduction about this yesterday, but when it comes to the genetics of dementia, the short summary is that it's complicated. We do know a lot in the sense that over the past few decades, we've identified more than 270 unique regions in the genome that associate with one or more neurodegenerative diseases. But for most of these regions, we don't really know why they associate, particularly we don't know the causal genetic variants. So just as an example, what I'm showing here on this slide is the latest Alzheimer's disease GWAS study involving nearly 800,000 individuals. And all these lines that you see represent a region in the genome that associate with Alzheimer's disease based on where they're located in the genome and then the strengths of the association. And the thing is that if you were to just zoom in into one of these genes, you're not going to find a single causal variant that leads to Alzheimer's. Instead, you're going to find that there's actually a cluster of variants that are all equally associated, which is represented by this cluster of red dots, each representing a different SNP. And you can see that this cluster of SNPs actually precisely corresponds to this one gene, which I'll talk about more in details later on. But what this actually means, the interpretation of this, is that there are actually common versions of this gene that are inherited into the population. So people inherit different versions of these genes. One of them, which actually increases your risk for Alzheimer's disease, we call that the risk haplotype, but also another version that decreases your risk for Alzheimer's disease because it has the opposite set of variants. And this pattern of risk and protective haplotypes, we see this in many of these GWAS regions. We also see it's not specific to Alzheimer's. You see this also in other GWAS studies. And so one of the current challenges is to try to understand what exactly is it about these haplotypes that drive these phenotypic differences. Yes, now what you don't generally see in GWAS study and also in the downstream characterizations are structural variants, right? So I think by now we know that structural variants are important. They can have very big effects. But historically, we mostly only focus on small variants, not so much on the structural variants. And so one of the things that we've been interested into is to try to understand whether some of these haplotypes, where they actually differ by structural variants. Because at the end of the day, if that is true, then these structural variants are actually important genetic features that we need to take a closer look at, especially given that some of these haplotypes do lead to either a neurodegenerative brain or a healthy one. And so for quite some time, we have been wanting to generate some long-read sequencing data of a case control setting to get a better understanding of structural variants in these known haplotypes, but also in general to start to associate them in sort of novel analysis. So we work very closely with the Alzheimer Center in Amsterdam. So that's actually one of our focuses. So for us, it's easy to get access to Alzheimer's patients, so we can generate data from these individuals. But still, that means we need to come up with a control group. And generally, in this type of case control setting, what you normally do is you get age-matched individuals who don't have dementia, and then you consider that you're healthy control. But if you consider these two cohorts as part of a spectrum of cognitive health, where on one side you have individuals that are demented and individuals that are non-demented, our point is that this spectrum doesn't actually end here, because there's a completely opposite end to this spectrum, and that is a very specific and unique set of individuals, which we call cognitively healthy centenarians. These are people that are at least 100 years old, but somehow managed to escape dementia. And that includes people like Ms. von Onderschipper, who at one point was the world's oldest living person at 115 years of age. Even at that age, she was still very cognitively healthy. They did neuropsychological tests on her. Her brain functioned like that of a healthy 60-year-old. She donated her brain to science, and when she passed away, they did the standard pathology examinations. She didn't have really any signs of neurodegenerative pathology. And so what Ms. von Onderschipper and other centenarians shows us is that it is possible to get to these extreme ages without dementia, but of course, the big question is how. So over the past 10 years, we've actually been putting together the 100+ study, where we actually try to recruit cognitively healthy individuals across the Netherlands. You can see some of these individuals on these photos on the right hand of the slide. And just to give you a couple of numbers, so at the moment we have 466 individuals in the study. The oldest living person is 109 years of age. And just a reflection of how cognitively healthy they are, more than half of these people still live by themselves. I mean, we still, I think if you go on YouTube and look at one of these centenarians, even like a few years ago, some of these are still biking on the streets of Amsterdam, playing piano recital with their friends back in their retirement houses. So these people really are quite unique. And what we do with these centenarians is we do neuropsychological tests to try to get objective measurements of their cognitive state, and then we try to do multi-omic measurements. We've already done proteomic measurements on the brains of some of these individuals. We're gearing up to do also transcriptomics, and what I'm going to talk about today, and also one of my focuses, is on the genetics. So as I just mentioned, for the past few years, we've been doing long-read sequencing of 250 Alzheimer's disease patients and 250 cognitively healthy centenarians from our study. We did this with the Sequel IIe, and of course, this is a lot of support from PacBio, but also from Radboud, so we're very appreciative of that. And just to give you some numbers, we've sequenced more than 1,000 smart cells, about 2 smart cells per sample. We're storing more than 37 terabytes of data, and that's because we actually keep the non-HiFi data for the same reasons that Alexander talked about yesterday, and I'll talk just a little bit about that later on. And on the bottom right here, what you're looking at is some of the numbers from the sequencing data. So every dot is a genome. Red is centenarians, blue are, red are Alzheimer's patients, blue are centenarians. The x-axis is a whole genome coverage, y-axis is a median read length, and so we're clocking in at about average coverage of 15x and average median read lengths of 15 kb. So we're busy with the data analysis, but I'll just give you some snapshots of some of the stuff that we've been seeing. And that is that so far we've identified more than 100,000 structural variants across all these individuals. And when you start to zoom in, you do see quite some common themes. One of them being that tandem repeats are major sources of structural variations. I think we've had already a very nice discussion about what these things are. They can be very variable, both in length and also in sequence content. But I think what's particularly important is that they can have regulatory properties, right? So if you have structural variation that involves these tandem repeats, you might actually be dysregulating the local regulation of genes. What we see is that these things are actually highly multi-allelic. I think actually at least 50% of all the structural variants that we find are from tandem repeats. And on the plot on the top here, what I'm showing you is how multi-allelic these tandem repeats can be. So generally, if you look at a structural variant that is not a tandem repeat, for example, transposable elements, which I'll talk about in the next coming slides, you can see that most of the sequences only have 2 unique allele sequences, which is one on the x-axis, meaning that for one location in the genome, you generally have one or two different, 2 allele sequences. But in green, these are tandem repeats, meaning that when you start to look at structure variation from these regions, you don't just have 2 allele sequences, you actually have many more. Right? And just to go show that basically the reason why there's also so many multi-allelic sequences is because of the fact that for a lot of these individuals, you have variable lengths and also variable sequences. And these things can also be pretty large. So far, we've seen that these distribution of these tandem repeats can be anywhere from a few hundred base pairs to several thousand. Now, I'm going to just quickly go through this, but I just do want to just say that we have started to do associations of tandem repeats, not just in the haplotypes, but also novel associations in Alzheimer's. You can read our initial findings in at least this preprint here. I'll talk about just one tandem repeat, just for the interest of time, and that is a tandem repeat intronic in a gene called OPCAT7. Basically, it's a fairly large tandem repeat. It has a 25 or so base pair motif sequence, and we see sequences ranging from a few hundred to over 10,000. And in a paper from a couple of years ago, it was proposed that this tandem repeat is actually associated with Alzheimer's disease, such that Alzheimer's patients are actually enriched with a version of this tandem repeat that's more than 5.7 kb. So one of the things that we wanted to do was to see if we can actually validate this suggested association. And the short story there is that, for reasons that have been already talked about, these types of tandem repeats can actually still be challenging to genotype. So what we saw is that, or actually what we did, is we took a couple of centenarians. We did a Salazer blotting assay to get experimental validations about the size of these tandem repeats. And then in parallel, we sequenced them on the Sequel IIe. And then we compare the lengths from both approaches. Basically, if you only use the HiFi data, you actually don't get much concordance. And that has to do with the fact that even though these were, even though these genomes are actually quite generally high coverage, ranging from 15 to 30x, when you go to this region, when you go to this gene, you just get very dramatic drops of coverages. We start to lose haplotypes, we start to lose reads, which leads to misgenotyping. But as Alexander talked about yesterday, if you start to go to the non-HiFi data, which has all these missing reads, then you can actually better assemble these sequences, and you can go back to concordance, so you can actually have the expected sizes that you expect to see. And this actually starts to propagate as well across all our genomes. Specifically, what we started to do is we wanted to see if there were indeed significant differences between the two cohorts. So on the bottom here, every dot is the size of a tandem repeat in log2 scale. So the closer to the right, that's the larger versions of tandem repeat. On the left, that's the shorter versions. If you only use the HiFi data, we don't see a significant differences. And if you go in the preprint, you'll see how there's a lot of dramatic drops of coverages, also high rates of homozygosity. So that really goes to have these red flags that we're really missing sequences. But when we start to integrate the non-HiFi data, then we actually start to get the separation of the two distributions, particularly that if you look at the upper and lower quantiles, we do start to see that, for example, Alzheimer's patients are enriched with larger versions of the tandem repeats, while the centenarians are enriched with shorter versions. Now, another major source of structure variation are transposable elements. I think this has also been already touched upon by previous speakers, but just to make sure we're on the same page, what these things are, that they're elements in the genomes. When once integrated, they'll try to be transcribed into mRNA molecules, and then we'll use a reverse transcriptase to try to insert themselves somewhere else. What we see is that at least a third of the structural variants are actually transposable elements. And when you start to look at what these things are, the fairly young evolutionary recent events, a lot of them are actually human specifics. This are things like the subfamilies from the Alu elements, SVAs, line elements, which are ultimately just a representation of how the human genome has actually changed over the past million years. And much like tandem repeats, these things are not junk DNA, right? They actually can have regulatory mechanisms. There's a lot of nice characterizations about the fact that they can influence all sorts of alternative splicing, gene expression, polyadenylation, and many other things, which of course means that if we start to talk about structural variants involving tandem transposable elements, especially those that land nearby separate genes, then they could also lead to disruptions in the local gene regulatory mechanisms. So I'll talk about just one very specific transposable element that we found. You can read it in this preprint that we have in medRxiv. And it's about a retrosposon in the gene that I'll talk about in a second. But I like to tell this story because it really just goes to show how we can use long reads to not only look at the structural variants, but also look at the methylation patterns. And as I just mentioned, also the ancestry, how that actually differs across different populations, and how that could also lead to differential risk, genetic risk for Alzheimer's. So very quickly, this gene, TMIN106B, it's a bit of a hot topic at the moment in the neurodegenerative field. We don't know everything about it, but what we do know is that it seems to be very influential in the morphology and activity of the endolysosomal system, which simply is analogous to our garbage and recycling center of cells. And actually just very recently, only in 2002, we started to realize that this protein actually aggregates in the brain. We see this in Alzheimer's disease patients, Parkinson's patients, frontotemporal dementia patients. We don't know whether these aggregates are toxic or whether they're actually a reflection of some larger neurodegenerative process. But what we do know is actually the genetics of this. This is actually the same gene that I was telling you about earlier in the introduction. Because already for some time, we've known that this gene actually associates with different neurodegenerative diseases. And as I just mentioned, we know that there are actually 2 common haplotypes in the human population. And it's actually the risk haplotype that is associated with increased aggregation of this protein. Now, we've also known for some time as well that there is actually a coding SNP that differentiates the 2 haplotypes. And for a while, people thought that this was the leading reason for the phenotypic differences. But there's been some functional characterizations that reignited the debate whether the SNP is actually doing anything or if there's other variation in the vicinity that might be also leading to these differences. Now, the short summary is that we have actually found structural variants, and one of the specific ones that we came across is actually a structural variant in 3' UTR of this gene involving what's called an oligoYBA element. This is actually a human-specific event. We don't see it in the primates or in the great apes. And at least in Europeans, this is in complete linkage to equilibrium, meaning that you only see this retrosposon popping in whenever the risk haplotype is present, but never in the protective haplotype. And so now the question right now in the field is to figure out, well, how much of this differences between 2 haplotypes is due to the SNP, the retrosposon, or maybe even both. But one of the things that we can actually do is to at least see what this retrosposon might be doing with the data that we generated already. One of them is on the methylation profile. That's because you have to imagine that if we let these retrosposons just propagate over and over and over again, that's ultimately going to lead to genome instability and cell death. And so one of the things that our cells do is that they come in and they methylate the retrosposon to prevent from this process from happening. But also there will be a pressure over time to also then end up with methylation patterns in the nearby vicinity of the gene. So one of the things that we wonder whether we would then see higher methylation levels in the risk haplotype whenever the retrosposon is there. And that's actually what we saw. So up here, this is the canonical transcript of this gene. And over here is actually the methylation profile of all 500 genomes, where every dot is a CpG island, and it's showing you the amount of methylation with the 95% confidence interval. And basically, what we find is 39 significantly differentiated CpG sites, such that for the haplotypes that have the retrosposon, it's more or less always methylated. If you're heterozygous, of course, you're going to have only one version that's methylated. And if you don't carry the retrosposon, then you don't really have any methylation signal. I'm just going to quickly skip to this because I do want to talk about something else. But I just want to say that we've also, of course, methylation profiles can be very tissue-specific. So we have actually sequenced the brains of some of these centenarians. Not many, but we do have for 10 situations where we sequence both blood, brain, but also some of the children of these centenarians have actually participated in a study, which means we can start to at least look at not only tissue-specific differences, but also age-related factors. And then, well, the short summary here is that we do see brain-specific methylation patterns. So this is the same outline, now just, or the same layout, just split out between the 3 genotypes. And specifically, this line in blue here, that's the methylation signal in brain. And what we see is that actually in this particular region, we do see demethylation in the brain. Can this lead to functional consequences? We don't really know, but we are looking at actually transcriptomic data to see if this might actually have an influence in alternative splicing, for example. Now, the very last thing is about ancestry. So what I'm showing here is the location of all these centenarians and where they come from the Netherlands. If you also do the same thing for the Alzheimer's patients, you'll see the same map. But as much as we would like to think about it, the Netherlands is not the global human population, right? Maybe we can get away with saying that it's a nice proxy for European ancestry, but it's definitely not for the diverse genetic ancestry that we see across the world. And the reason I say that is that if you only look within our cohort, of course, you will see the presence of these two common haplotypes. But when you actually start to integrate other diverse genomes, for example, from the Human Pangenome Research Consortium, what we actually already see is that even just looking at the SNP frequencies and the correlations between them, is that yes, these haplotypes that we've known of for a while are there, but so what it looks like is actually other haplotypes. And specifically what we see is that there seems to be other haplotypes specifically in subpopulations of individuals with African ancestry, which of course, if we know that these are actually leading to differential risk for dementia, what exactly these things are doing, well, we don't really know since we never actually looked at them. And that's, I think, where HiFi data actually comes very in handy because, well, you can actually start to do this. You can phase SNPs, you can and structure variants together. You can define the haplotypes, and as a sort of a population framework, you can actually look at how these things actually compare to each other. And so what we did is we actually took the haplotypes from our cohort. We looked at the Human Pangenome Research Consortium haplotypes, and what we actually found is that indeed there are novel haplotypes in these individuals. Just very quickly, this is a bit of a busy plot, but what you're basically looking at is a binary heat map where the columns are every variant that we find in this gene. So it could be SNPs, it could be structural variants. But basically, when you see gray, that means that that variant is there. White is absent. Every row is a haplotype. And the colors corresponds to either whether European, which is orange, or African, which is green. And what you can see is that if you start to compare and cluster these haplotypes, you find back the known risk haplotype and the known protective haplotype. But here in the middle, you'll see that there are these two haplotypes that are predominantly present in African individuals. And what actually is happening is, if you'll notice, that there's actually what seems to be a meiotic recombination, such that variants in the 3' end are now actually separated because the variants in the 5' end and the 3' end are now in two different haplotypes. And what's interesting is that in these particular individuals, the SNP, the coding SNP, and the retrotransposon are now completely separated. And at the moment, if you just go and look at what people are trying to do, they're trying to generate mouse lines, iPSC lines, and then try to use CRISPR to go in and change the SNP. But of course, you could also use these sequences because the setup to try to figure out what exactly are the functional consequences of the SNP that are retrotransposon, what's already present in these individuals. Now, all this, of course, was, at least that last story, was only in one gene. And I think what we're ultimately seeing is that there's really complex evolutionary histories for all these different regions, for all these different GWAS locations. And so that's, I think, part of the challenge to actually try to digest all the information. But just to conclude, as I just mentioned, we recently finished the sequencing of all these genomes. I talked about the fact that a lot of these structure variants are coming from transposable elements and tandem repeats. And of course, with this data, we can start to look at structure variant discovery, the methylation differences, and ultimately also the integration of other data sets, which I hope to also see more and more, especially from the diverse ancestry sections. Just to want to acknowledge the group. This is, of course, a collaborative effort. Especially acknowledge Hanna Holstega, who's actually the head of the study. And with that, I'd like to thank you for your attention.